Okay, we are live and lively with Speed of Sound and Walking Fish and Immutable, or Andy today. You have a new name. And you want to introduce yourself, Demeter? You are the guest.
Yeah, so I'm working as a molecular biologist, and I'm, let me say, professional scientist doing real laboratory experiments for cancer research. And I've done that very, very, very long time. So, but I will start after when everybody is introduced, I can start my story, how I come to the origin of life, actually, topic. It was very interesting story, actually.
Oh, they all know us unless you two want to say something. So, no? Yeah. Yes?
Well, I mean, does Demeter want us to introduce ourselves more or less?
Oh,
for
yeah.
him?
Okay.
Or for...
Yeah.
Okay. Well, just more or less, I am an evolutionary biology undergraduate right now. I am from rural Alabama, and I grew up fairly creationist and holding a lot of beliefs that I don't anymore. And that's part of the reason I started this, and just because I like talking about biology. I guess my family gets enough of that, so might as well have another source for me to dump all of this information out to.
Is your parents also creationists, or they're just...
Well, my dad's not. My mom is she's like on the fence about it. I think I was more into it than anybody else in my family was. I think my brother is as well. It's not usually a pretty big topic in my family, but my family is very religious, like all around. So
yeah, it's understandable.
Yeah,
it is understandable. So the origin of life is the final frontier for the creationists, because this is the foggy, muddy area where nobody knows how it happened, and everybody speculates everything that he thinks it's a correct thing. And of course, this is a wonderful kind of media for creationists to provoke, to say something about intelligent designer or something like that. So I can tell you guys how I came to the interest of origin of life. So approximately, I think was maybe 4 years ago, something like that. Just I was so bored and just browsing YouTube, interesting topic, and somehow I come to lectures of creationists.
Mm-hmm.
So, and I started to chat with creationists, and always in that case, it's disputes became. Everybody's have his own arguments, and in this discussion, I think was around Discovery Institute guy, which I think it's Stephen Mayer. Probably was his name, from Discovery Institute. His theory, I mean, you're supposed to know what is Discovery Institute, right?
Yeah.
It's not necessary to say what the Discovery Institute is. This is pseudoscience institute.
Mm-hmm. It's an idea proponent.
Yeah.
Yeah, they are quite shady, let's say that.
So, I mean, from the perspective of someone that is working hard science, experimental science, in molecular biology, I can tell you now why the people switched and go sometimes in a Discovery Institute. The reason is very simple. The science is very difficult area, hugely difficult. You constantly depend on grants. You constantly have to prove yourself. You constantly have to produce results, and it's nothing sure. You're constantly in a not sure situation. So I'm not sure what I will do as a scientist after, for example, 6 months, because the funding is going out. So, and this is a very, very difficult. Guess what? If you go in Discovery Institute, what will happen? You will have a wonderful salary. You can lie all the times and nobody's care. And you'll enjoy a wonderful life. Everybody is happy. Everybody will be happy. You'll be happy. Your family will be happy. You'll have your own society. And that's it. But you sacrifice your scientific conscience. So that's why some scientists, actually real scientists, like James Tabor, I think he was actually a real scientist, chemist, and he went to Discovery Institute. So that's what actually it's all about. And I don't think the Discovery Institute is care at all about what the science think about evolution. That's not the function of Discovery Institute, to provide any truth about the science. So the function of Discovery Institute is just to reinforce the believing of already creation is created. And they know very well that they cannot convince anyone in a real science field. So this is about the Discovery Institute. Anyway, so I don't want to talk too much about this because we have lots of ground to cover. Now, what happened during that time, Stephen Mayer, actually, he was, around 150 area. Thank you very much. So his kind of concept was, if you have information, that means intelligent designer, based on computers and information that we know everywhere. So that was all about his lecture. And I started to argue that you can create actually information without having intelligent designer, just because you can have a process of selection. Of course, for Discovery Institute guys, there's such a things like evolution and selection does not exist at all. So, and we argue, and so on. I think was some of the classical Gneiss was her name. What was her name? Something like that. Anyway, it's not important. What is important is, after this discussion, I started to think about actually what might happen and how the life originated. And these discussions with the creationists actually triggered something in me, and I started to think about constantly. Even I found myself that I cannot stop thinking about it somehow. So it started as a process of just something came out in my mind constantly about origin of life, how it happened and what it happened. So as a professional molecular biologist, how the RNA works, how the protein works, how those kinds of things, I can imagine all of that. So to me, it was not so important at that moment to go and look what actually is published in this area. So without reading what is published in this area, because this is not my area of research. So by thinking about, I actually it took me a while, of course, but I came with hypothesis how actually is origin of life come to be. And in fact, after that, when I formulate my hypothesis, after that moment, I started to read the literature and started to look what the others colleagues in the field that actually are paid to do that are actually published for the origin of life. Now, at that moment, when I started to read, I was just expecting to see my hypothesis immediately in the next paper. When someone should formulate exactly, although it's not with the same wording, the same things, should formulate the same thing as mine very soon. So I was starting reading first paper, second paper, looking RNA world hypothesis, all kinds of hypotheses. And suddenly I realized that whatever was in my mind is a hypothesis. It's not available, it's not published. And something else, everything that it's published, it's completely wacky. It's simply for some reason, does not explain everything that I want to explain. So that was the reason I stopped myself at some point and started, actually, I became very serious about it, and I said, okay, what's going on here? I mean, look at that. I have a hypothesis which have a huge explanation power. You can explain almost everything in the origin of life. It's not published yet. And at the same time, everywhere, the people from Harvard, Jacques Zostek, Kunin, Nick Lang, Bruce Damon, and many others, everybody is publishing. Those people are supposed to be the guys which supposed to lead the area, and none of that hypothesis is relevant and have this explanation power. And of course, I called to a friend of mine, which it's my buddy from back in my country, and I told him, let's publish this. After all, we have to publish it because it is very important. And that's how we started to publish. So it came the first paper in 2018. After that, I have already idea for all the papers and all the 3 papers, but I didn't publish everything at once because I was busy. And you can guess this is not my area that someone will pay me for that. So I started to publish one by one when I have time. 2020, this year, I published the second paper. So the first paper was the hypothesis, the major hypothesis. The second paper, which we published this year, it's all about the origin of life, of course, and the origin of genetic code. Mm-hmm. And of course, I will tell from everything a while. And I'm prepared right now. I have the manuscript for the third paper, which will describe mostly what actually the life is, because this is a misunderstanding totally what the life is. At the moment, right now, if you think about origin of life research, and if you look the professional origin of life researchers, this is several major centers for this. The bigger center is Harvard at the moment, and California, of course, there is another one. Some in Europe as well. And I'm calling this period of time the big talking. Everybody is talking for origin of life. Everybody considers himself as the correct one. Yeah. And everybody wants, you hear him in a lecture or anything like that, he behaves like exactly he knows exactly what happened. The confidence of any speaker in this area is a huge. Just recently, I was watching a lecture from Nick Lane. He's the metabolism first hypothesis guy. He behaves that he knows exactly what happened in the area. Jacques Zostak is the same. You can listen him very well, and he's also the same story. You can see him. He's so confident. He knows exactly what should be, how should be, and so on. But I can stop here in the introduction period. So in introduction, I can start now to talk a little bit more the more important things. So because that was my story of how I was involved in Origin of Life. It was very interesting to think about. Someone comments, any questions up to here? Because
well, are you still doing like cancer research and stuff, and just you do abiogenesis as a side project, or
what is the that's so at the moment after COVID-19, I'm in a difficult situation in my cancer research.
Yeah.
So in the current lab, I've been probably 12 years in my current cancer research lab in Roswell Park Cancer Institute. There is no more funding until the end of August. Of course, I'm working also with University of Buffalo in the laboratory, again for sort of cancer research, but more specific to the replication, DNA replication.
mm-hmm.
And this will continue for a while, but I'm not sure how it will continue and to what extent. So, in fact, I will apply I will submit a grant sooner, probably until the end of the year. And who knows, I might be out of any job. That's how the actual science is today. So I should go in Discovery Institute, maybe.
I was thinking about it. If I were a good liar, I would definitely apply over there.
Yeah. Exactly.
All you have to do is sound like you know what you're talking about.
Exactly. Yeah.
And you got it. And I'm good at convincing people that I know what I'm talking about, but I sometimes don't know. So, do you want me to share one of your papers on the screen, or?
Oh, you can post links to my papers, 2 papers.
I did, but I can actually put them on the screen so people can see.
Maybe for a while, it's okay, no problem. So
the 2018 paper, you want that one?
2018, it's a good. Yeah, so you can put there.
Let's see if I can find it. I couldn't have 2018.
So,
so there it is.
Yeah, this is the 2018 paper.
Yep.
So
if we could show an illustration or something if you want to, but I'll let you go ahead and figure out how you want to explain this.
So I will come to the paper, of course, but first I have to say you can remove at the moment. Actually, we'll post for sure. But let me explain a few things, which is very, very important. First, if you think very serious about origin of life, any hypothesis for origin of life is supposed to solve several problems. The first problem, of course, it's so-called chicken or egg dilemma, or chicken or egg problem. This is what comes first, RNA or proteins. And this it's one of the problems which it stay constantly in the field and misunderstand the people. Constantly they are confused by this problem. I will show you later that such a problem does not exist. Simply, there is no such a problem. The second thing that's supposed to be solved from any hypothesis for origin of life is how is how the ribozymes was selected, because there is a natural ribozymes. And this is a problem, and I'll tell you why it's a problem. Because the ribozymes are enzymes, which is RNA enzymes. Usually the people said that they are not very good enzymes. The ribozymes are not very good enzymes, and that's why they just was left over behind and was overcome by the protein enzymes. This is not a way of thinking. The ribozymes actually could be very powerful enzymes. And we know today about the ribosome, which is in fact ribozyme. And this is the most powerful and the most productive enzyme ever in the universe, maybe. It is ribozyme. So the peptide bond formation in the ribosome is a ribozyme. So you cannot think about that something, it's more like a powerful or more willing to become enzyme than the other. Since the selection for start, for some reason, the protein enzymes became more dominant, and of course they overcome the enzyme work. But how that happened in the origin of life, and this is a huge problem for any hypothesis to solve it. Because if you have no idea why, in that case, ribozymes was selected, you cannot solve the origin of life. The third now problem that any hypothesis is supposed to solve is the origin of genetic code. If you don't have answer how the genetic code started, you don't have any hypothesis for origin of life. And of course, one of the other, I mean, there's much more things to be solved for sure for origin of life, but one is the energy, so-called energy. Very frequently, us molecular biologists was blamed that we always are talking for RNA, for proteins, for those things, but we don't care at all where the energy comes from. So, and this is usually a blame from the guys which are in a metabolic first hypothesis. So this type of solving of problem must be, no matter what, must be in any good hypothesis for origin of life. If you don't solve all of that problems, you don't have good hypothesis for origin of life. Of course, this is include how the information came to be and how this is part of the genetic code. Now, at that moment, I can tell you the most wrong thinking in origin of life. And when I'm saying this, I'm saying in a way that, of course, this is my thinking. This is my thinking, but I'm sure in this my thinking, I can defend my thinking very well. And because of the huge explanatory power of the hypothesis that I have, I think this is the correct way. So the very first misunderstanding in origin of life, and remember this is a huge misunderstanding or wrong thinking in origin of life. And this is to say life started, and after that evolution kicked out and start to develop new forms, start to complicate new forms. This is a huge misunderstanding and huge misrepresentation and wrong thinking in origin of life. That's not true. You cannot think that life started and then the Darwinian evolution or the evolution kicked out. Simply does not work in this way, because the life by itself is a Darwinian evolution. You cannot separate life and Darwinian evolution from each other. This is the same phenomenon. It's the same thing. If you start to separate it, you are in a wrong way. There is no way to have life without Darwinian evolution and vice versa. If you have Darwinian evolution, you have a life as well. And that will be included in my third paper that I'm going to submit until the end of the year. So, what actually is Darwinian evolution and what is the life? Obviously, it's the same thing. If you have some information source, and this information source determine structure by some rule. So determine and predispose, define structure from the other side. And this structure at that moment could stabilize the information source. So you have information and structure or function. Usually the biologists, the classical biologists, call this genotype and phenotype, of course. But if you think about in a molecular level, you still can have source of information like RNA. And the RNA, by using a genetic code, determine the protein sequence. And by determining the protein sequence can interact with RNA and actually protect it. So what you have a system where the information determines the structure. I would like to know that everybody is on the page, right? Everybody understands what I'm talking about.
Yeah, so you're basically saying like prebiotic molecules still would have undergone selective pressures, and the ones that were the best at
I will come to that.
I do want to ask a clarifying question. Do you think Darwinian evolution or natural selection rather can act on like chemical systems, like in terms of chemical reactions and thermodynamic and kinetic barriers?
I will come to that as well a little bit later. So we'll discuss just a second left.
Yeah.
So at the moment, just what is important to know that if you have a system where the information determines a structure by some rule, some sort of rule, in our biological system, this rule is called genetic code, or could be Watson-Crick base pairs as well. It's still a rule for determining a structure. If you have that system and you put this system in the environment, it doesn't matter what kind of environment you put the system, what will happen? One thing that will happen is this system will perish if the environment is very harsh, or will be selected, will survive, but will survive because of the structure which harbor and preserve the information. And this is how the Darwinian evolution works. You have information which determine the structure, and this structure is preserved, preserving the entire system. So what will happen if there is a mutation or changing of information, because there is a rule for transferring to the structure or function, this changing of information will go blindly to a new structure, completely blindly. So the new structure will be changed, but that, of course, will be subject of another selection of the environment. In that case, you always have a step which, if something survives, will remember the previous situation, the previous system, the previous stage, and whatever it's happening. This is the only way you can have increasing the complexity of the structure. Without this, there is no way to have complexity. Origin of life is exactly that. The beginning of information, structure, and the rule between them. Otherwise, you don't have life. You don't have anything like that. So I can immediately say what the life is. The life is dynamically stable structure where the information determine its own supporting structure, dynamically stable system where the information determine its own supporting structure.
Right. So the stabilization is a feedback from you go from information source to structure, and then feedbacks into the information source. And
so essentially, what's being described is that these chemical systems are adapting and responding in order to replicate according to whatever environment they find themselves in. Is that correct? Or
exactly.
Yeah. So Otangelo was asking. Go ahead.
Think about this particular system. At the moment when you have the beginning of this system, you have situation where you can start making more complex, more complicated, and more adaptive system because of the feedback that you mentioned. You have always feedback, and this feedback, it's not just a feedback. This is a feedback which is determined by information. It's not just a feedback by itself. So if you don't have connection between information and structure, even that you might have information, even that you might have another structure, that doesn't count at all. You have to have feedback in that case.
Yep.
Mm-hmm. So how would that feedback loop begin to develop, right?
Well, that's a stabilizing part, isn't it?
That's my hypothesis. Yeah, that's the important part. Yeah. That's the important part.
Well, I think what's going to become interesting as well is that, for instance, you might have some systems which are chemically very stable, so some systems which are not chemically subject to modification very easily. But in terms of making it replicate or trying the molecules to reproduce on their own based on whatever catalytic agents you have, might be very difficult and unfeasible as a system.
Exactly. So that is not life, meaning it's a totally separate system. For example, if you have, there are lots of complexes, chemical complexes. There is a polymers which can become very, very, very big complexes. Sometimes you have information somehow, but without any feedback. Let me say growing crystal. If you have growing crystal, you have information there because the crystal it grows, but there is no executable structure in that, and also this is not dynamically stable system. So the rule, I mean the definition of life, and the same time the definition of Darwinian evolution is the same. Dynamically stable system where the information determine its own supporting structure. That's it. That's all about. Now, again, I'm saying something from the third paper that I'm going to submit, but there is another thing you have to consider. Now I will submit and provide a new law. This is a law for origin of life. The law of origin of life, it's something very important, but it says that the information, the corresponding structure of function, and the rule between them are locked in its origin. You cannot have information to start and to wait for some structure to appear. They are locked in its origin, meaning they should be originated in the same set of processes. Simply because there is no way to determine and to have information without this information to execute a specific function, specific structure. Otherwise, this information will be random completely. There is no formulation of information without having executable structure.
Okay.
And to do that, you need a rule. Without rule, you cannot make it.
Mm.
So the law for origin of life is that the information, the structure, and the rule between them are locked in its origin. There's simply no other way.
Mm-hmm.
So any hypothesis for origin of life is supposed to provide mechanism how that happens. Otherwise, it's not possible.
So are you basically arguing that like the whole idea of metabolism first versus information first is a false dichotomy because you have to have these two processes working like parallel to each other?
That is the problem of metabolism first hypothesis, because those problem they cannot explain this informational part.
Yeah.
So they usually explain source of energy. They explain very good prebiotic synthesis very well.
Mm-hmm.
And they said it start to assemble aggregates or start to assemble some complexes and so on. This somehow cannot happen without information. As I said, you're supposed to have Darwinian evolution structure, as I mentioned, information and structure together. Otherwise, the system will not work. It will not advance.
Okay, so one fair critique somebody could ask would be, is it fair to say that the information and structure are reliant upon the energy? For instance, in the case of replication, typically, you need a very wild host of enzymes to unwind a double strand of DNA and to begin replicating and adding nucleotides to the strain to be preserved.
I understand, but that's the trick. This is the moment of discovery. You have to figure it out how the very simple system started, because if you might have you can create this system in a laboratory condition,
mm-hmm.
but you will create a very complicated system in that case, and that will be just artificial life. You can make it, but you have to figure it out how it's supposed to be very simple, because if it's not simple enough, it will be statistically improbable. And if it's statistically improbable, you don't have good answer. By the way, I can tell you right. Yeah.
Oh no, go ahead. Go ahead. I was going to ask if you think
about
protocell. I'll go ahead. So, do you think protocell or like Luca type systems would have to use replication that could be obtained, like using energy from free energy in the environment already, that it needed a metabolite to replicate? Or
so I will come immediately after that. So I was just talking about the first misunderstanding of the origin of life, and that was that life will start and after that will kick out the Darwinian evolution. This is wrong thinking. You don't have that case. You have directly Darwinian evolution. The beginning of Darwinian evolution is the beginning of life. There is no other way. This is the same thing. And everybody, and unfortunately, among the professional scientists, they simply do not understand it for some reason. That's why I'm going to submit the third paper for that. Now, just give me a few seconds. I will be back, go to the bathroom for a second, and I'll be back and continue with the second misunderstanding, and that's about the replication. Misunderstanding or wrong understanding of origin of life. Just give me probably 1 minute.
All right. So in this little intermission, should we talk about anything Otangelo has been saying, or should we talk about his paper?
Do we want to block Otangelo? I mean, he's going all over the place in the chat.
Nah, let him go. This is the paper that he's talking about with James that I downloaded, the implausibility of metabolic cycles on prebiotic Earth. So I just thought I would download it and display it. This is from 2008, Orgel or something. But anyway, I thought for RJ's, well, RJ's probably looking at the paper. But anyway, Otangelo quoted this part. But yeah. I don't know.
Well,
it's up to you. It's up to you. If you vote to block him, I will. But
no, I don't think it's realistic.
No, I think Otangelo is pretty cool. He does go on. Carbon dioxide.
What was I gonna say?
Okay, so we have the the first problem, the idea that that Darwinian evolution kicked off later after all this, which was my thinking, and saying that that's the first myth, that basically the selection was there from the beginning. But I have a little diagram here going from the information source, which could have been very, very simple, to the to some structure, and I think they talk about bridge peptides. And that basically you would have nucleotides with bridge peptides, and that would stabilize the information source, and it would go around and around and become selected. But the interesting thing is, like, okay, I'm a programmer, computer programmer. When I develop, like, if I'm given the task of developing some fairly simple computer program, the first thing I do is put down a very, very simple 3 lines of code, a little loop, where I can like read the file. And then I go back and I do some selection, and I say, okay, no, I got to do this because we want this output. And then I'll do this, and then all of a sudden it fails. So I cancel that, erase that, and look for another solution. And I keep adding little tiny bits to it until I have a very complex algorithm. So it's kind of the same sort of thing. There are very, very tiny steps to creating any algorithm. And it works that way in computer science as well as anywhere else, except in computer science, I am the selection mechanism. So I'm the one that decides on the output. So anyway.
So is outcome, that's nice to say, because from computer point of view, and I was thinking you can create actually to make some sort of model in a computer how the selection goes. Based on this and this idea.
Yeah, you could develop, you could actually write another program that did the selection, but normally computer programmers are sitting there and they're saying it works or it doesn't work. The algorithm either dies or it works. And if it works, well, then you go and make another change and see if that works, and you keep going. And pretty soon you've got 2,000 lines of code and you have a very complicated program. And when looking back, you'll say, I have no idea how I wrote that. So,
yeah. And that, by the way, you can create in the same principle as I said, information and executable function structure. You can make it entirely on the computer in a program.
Yeah.
Of course, the environment will be different. The selection force will be different. You can create your own selection force. And in fact, you can make a self-developing program that you can select it to do specific things.
Yep.
And that's life on the computer. It is life.
Yeah. I agree. Yeah.
So let me start with the second huge wrong thinking in origin of life field. And this huge wrong thinking in origin of life think it's so powerful wrong thinking that it's embedded in the mind of the top scientists in the field. Do you understand how wrong thinking is? So much. It's called self-replication.
Okay.
Self-replication is the second huge wrong thinking, misunderstanding of origin of life. Have you ever, I'm asking everyone, have you ever seen natural self-replication system? No. There is no self-replication system. I'm consider myself a specialist of DNA replication and cell cycle. That's my field of expertise. There is no self-replication system. Never, ever. This is a wrong thinking, misunderstanding, and continue over and over again. You can see papers from high professional grades. They are making models for self-replication, presenting how self-replication could be, and so on. I don't think ever happened. There was not self-replication at all anywhere in the beginning of life, even now. So this is one very, very wrong thinking. Why is that? Why is such a misunderstanding or wrong understanding exist as a self-replication? Simply because of the RNA world hypothesis. RNA world hypothesis, in order to exist, you're supposed to have a stage where you have stable RNA, which stable RNA should develop itself into the ribozymes, which ribozymes should start self-replicated in existence. And that will work against the destruction, against the degradation of the RNA. It makes perfect sense from the RNA world point of view. However, I don't think that happened at all, simply because there is no way that was never. So think about this way. If something is very important at the beginning of life, it will stay important. You cannot replace it so easily. If something is embedded in the origin of life, it's very difficult to replace it later, because everything else will depend on it. So if you think about a ribozyme, which is self-replicating ribosome, what will be the force that will replace this very well-established self-replication ribosome? How the selection will work in order to replace with something different as a replication, as a replication. Right now, all the replication, nucleotide replication, like RNA or DNA replication, is performed by enzymes, protein enzymes. In fact, if you think about the actual catalytic center for any polymerase is only 2 or maybe 3 magnesium ions. That simple is in fact, this is so simple catalytic center of any polymerase. It's super, super, super easy to think about for beginning of replication of some short peptide that holds 2 magnesium ions. It's a several magnitude simple system to solve the problem with the replication than to think about self-replication.
It is important to note that for like organometallic systems like hemoglobin, for instance, it uses iron instead of magnesium, both of which are oxophilic, though, iron and magnesium. But the chemistry of those inorganic metals on their own is too highly reactive for species like oxygen. Yeah. It would usually cause peroxide radicals to form or things of that nature.
Yes, and that's why you still think that the origin of life is kind of anoxic condition, so you don't have much oxygen around during that time. So, but
or at the very least, you need something to stabilize or to kind of tame the reactivity of these metallic species.
Exactly, and that could be easily done by short proteins. You don't need to have a short peptides. You don't need to have something so complicated. So the problem for self-replication, it's another wrong thinking for the origin of life. And I'm surprised that it still continues. You will hear constantly from big scientists like Jacques Zostek, Bruce Dahmen, David Deman, Nick Land, many other scientists like Higgs, Paul Higgs, I think was his name. Many scientists thinking the same self-replication as something so normal, so well embedded, because the RNA world is so well dominating the field. So, and there is another problem here, because of this domination of RNA world, we simply it's very difficult to solve the origin of life per se, because RNA world is a system where you have to have many steps to come to the translational moment, where you have a translation as a process. So starting from short oligo RNA, short oligo RNAs, which supposed to stabilize themselves, and first of all, they have to replicate themselves somehow non-enzymatically. Non-enzymatic replication, which is okay. This is a process shown very well to be performed. Of course, it's mostly like GC, non-enzymatic replication, but you should have it. And after that, so they claim, so the RNA world hypothesis actually postulate that you must have very high fidelity replication, non-enzymatic replication. And this is the work from Jacques Zostek mostly, to have non-enzymatic replication in order to produce stable forms of RNA, which will be stabilized, and from them will be selected self-replication ribosome. Now, you can make everything from ribosome. You can make all kinds of activity as a ribosome in the laboratory. That's okay. I mean, I'm sure you can make life entirely based on RNA without any proteins. But I don't think this is the life as we know it and as it started. There are lots of problems there. And the most critical problem for RNA world is the transition from, let me assume you have a very stable, good, stable self-replication ribosome. Let me assume you come to this point somehow, magically. It must be a process of polymer transition in order to come to the RNA protein system, where you have genetic code. The formation of genetic code is very difficult to be provided from RNA world. I've been reading probably around 50 different hypotheses how that happened. The transition from RNA world, you have polymer transition to RNA protein world. It's a numerous hypothesis, huge number of hypotheses. The people spend fortunes with this. Publishing papers, who knows what. And there is a very clever ideas, actually, I can say. I mean, they have very good ideas for something, but it's not what we need. You need a feasible system to do that. Something very simple. So so in this introduction, I will stop with the misunderstanding of the RNA, of the origin of life. So RNA world, I just said there is a huge problems with the RNA world because cannot explain the polymer transition and cannot explain the formation of genetic code very easily. The selection forces are kind of very difficult to explain what the selection force will replace the ribosome that makes a genetic code to the actual proteins. to formation of the proteins. Since right now, I mean, today, all organisms have amino acid synthetase as the proteins which choose the right tRNA and right amino acid. In fact, the genetic code is embedded in amino acid synthetases.
Mm-hmm.
This is how it's somehow in the structure of the amino acid synthetases, it is the genetic code knowledge. So they know the only moleculars that know the genetic code is the amino acid synthetases. And now how you can explain the selection forces which come from the ribosome that determine pair between amino acid and tRNA to be changed from dramatically different system. There is no way to do that. I mean, imagine you build a house and suddenly you decided to make upgrade of your house and you start with demolishing the first floor, leaving the second floor on the top. How are you going to do that without crushing the entire house? So that is another problem of the RNA world hypothesis. Now, initially I was very convinced for RNA world hypothesis. Actually, to me, it was so natural without understanding, without knowing everything, because I was molecular biologist. To me, RNA world was so naturally, yeah, of course. But now that's not very useful. So I'm going to start describing actually the papers now directly, the papers, what is inside the papers. But before starting this, do you have any questions?
No.
Anyone to think about at the moment? So I told you about the definition of life, which is the same as the definition of Darwinian evolution.
Yep.
I told you a new law for the beginning of life, which is very important. And also I told you about the wrong thinking or misunderstanding of the field of origin of life. Unfortunately, this wrong thinking, it's so powerful, they will continue to be dominant in the literature. And I'm not surprised about this, because that's how the science at the moment is. It's a process. We'll come there. We'll come to this point. But I don't know when. It's very difficult to me to predict that. So the first paper that you see here, we published back in 2018. It is a paper which it's against every single hypothesis that you know to that point. And at the same time, there is a piece of every single hypothesis which is accepted in this paper. So every single hypothesis from one side is correct, according to this paper, and from another side is wrong, according to this paper. So what I can tell you, in a prebiotic condition, Earth, in a prebiotic condition, you can expect for sure a lots of prebiotic synthesis. Like it's already shown that you can expect to have a very short oligo RNA. Of course, this RNA, it's not necessary to be exactly identical to the modern RNA, but it's very close, should be very close. Of course, it's modified. This is open system. You can modify it. It could be anyway. Also, we know very well from many experiments that amino acids are presented. It could be presented in a prebiotic Earth. And also in a prebiotic Earth, you can explain and you can see very well, you can postulate and you can be sure absolutely there is a cycles about wet and dry cycles, hot and cold cycles. And this is a normal to think about the prebiotic Earth. You cannot have a steady state situation and life to originate it. It's very difficult to think this way. I can tell you right now the role of the moon, role of the moon in origin of life.
Okay.
During that time, during that time, moon was about 8 times closer to the Earth. So you can imagine sitting there. Sometimes you can just watching today's moon and just imagine making 8 times bigger. So how that will look like, it's cool. And the Earth was spinning much faster during that time. It's very faster. Probably the day for this prebiotic Earth was about 8 hours, something like that. So if you stay in the dark and watching the moon, this enormous moon, if you stay, you can see how the moon is moving slowly in front of your eyes, like that.
Mm-hmm.
So it's you can imagine it. You can imagine this situation.
Mm-hmm.
But what comes with actually the moon when it's moving around the Earth? It comes to things: tides, huge tides. During that time, the tides are enormous. Was enormous. There's no other way. So imagine, like Atlantic Ocean, every week to go and to flood the entire United States of America to the Rocky Mountains and back. So you can imagine such a huge tide, 100 meters tide, for example, or something like that. So in that case, the moon created a situation where you have wet and dry cycles. So one of the contribution in the field for David Diamond, his contribution was to show that in order to polymerize the RNA, you need wet and dry cycles. And that's a very good contribution to this guy. He is one of the kind of pioneers in this area. And he showed that you need wet and dry cycles in order to have a polymerization process. And that's completely understandable. So everybody, you know chemistry, most of you know chemistry, but to have polymerization, you need to remove water, right? And to have, so to digest in monomers, you need to add water. So polymerization process require removing of water between the two monomers. And that's why the wet and dry cycles are very important for that moment. That's why when you go and eat barbecue, and there are lots of meat, pork chops. A few hours later, you are very thirsty. You want to drink water. Why you want to drink water? Because you need water to digest the monomers. This is the same process of digesting the monomers. So you can go in the figure number 1 with the paper, Mike.
Go where?
Yeah, just go to figure number 1.
Figure number 1.
Yeah.
Okay. I'm trying to keep track of chat too. I've got a lot of plus yeah. I'm looking up papers. Who did you say was the person on the wet dry cycles?
David Diamond.
Demon?
I can tell. Just give me 1 second. I'll give you exactly the name. David Deamer.
Oh, Beamer. Okay.
No, not Beamer. Deamer.
Deamer. Okay.
Deamer.
DEA. Okay. I'm gonna put that in chat.
David Deamer.
Okay. So people can find it. Okay. So going down to try figure 1.
So if you go in a figure 1,
there we go.
Yeah.
Figure 1.
That's the figure 1. Usually it's referred as a prebiotic PCR. So
Okay.
who don't know what the PCR is?
The
Anyone that don't know what the PCR is?
I know what it is.
Yeah.
The peptide. Well, let's see. What is that? PCR?
Polymerase chain reaction.
Oh, okay. PCR. Okay.
PCR.
Gosh.
Now it's very famous because this is the PCR test for COVID-19.
Yeah.
The people can amplify is utilizing PCR. They can amplify any piece of DNA that you want to see what is there or what is not there. I'm doing PCR very frequently, every other day. So, but the main idea is if you put so here's the point. Here's the deal. When you're thinking about RNA world, when you think about origin of life, the very important notion to anyone that is working in origin of life is not to tell the Mother Nature how to make life. You have to set up your mind and completely eliminate your pre-thought, predispositions that you know how the life started. That's what happened with the RNA world. They always constantly tell to the nature how to make life. They tell the nature, you have to make ribosomes, you have to make self-replication, you have to do that, you have to do that. And they try to prove it. This is not the way how should think about. If you think about origin of life, you should eliminate everything and just think naturally. What actually could happen without telling the nature what to do.
Uh-huh.
An important kind of follow-up question to that is there are people who suggest that like amino acids, based on how they're generally characterized, at least alpha amino acids, you have an amine group, you have a carboxyl group, and you have an alpha carbon that has a hydrogen and an R group. And there's thousands of potential amino acids that could have been on the Earth potentially, and also in some of the not quite RNA world, but kind of replication first people who posit that hypothesis, they propose things like peptide nucleic acids or things like that that could be self-replicators. Do you think that there's potential for other, potential self-replicators?
This is a relatively natural way of thinking. So whatever you can see, for example, here on figure 1 is just simplified picture. In fact, everybody is aware, if it's serious about that, you don't have just amino acids. You don't have just lipids. You don't have just oligonucleotides. It's a mix of complicated, not just alpha amino acids. You have all kinds of amino acids. And you have very, very complicated environment.
Mm-hmm.
But whatever I'm describing here, it's something that is important for the next set of selection process. So what is important after all is what has been selected after formation of Darwinian evolution. That's why I started to tell you first about the Darwinian evolution. Yeah. In that case, it happened. Yes, there was many interactions. I agree, absolutely, many interactions with everything. But what is here is just what is come to be after the selection.
So that kind of raises
if you see this picture in figure 1,
that raises the other question that some people are still undecided on with the origin of life, is that are the building blocks we use, for instance, like the amino acids or sugars that we use as metabolic sources, could we call them unique in any sense? Like, or is this just a matter of throwing dice and this is one of the potential configurations that show up?
It just happened in this condition, prebiotic condition, and this particular setup of the primordial Earth It just that was selected. That doesn't mean in another conditions different things cannot be selected. For example, RNA should be selected, even that might not be exactly the same RNA that we know it today. Some sort of RNA with the same configurations should be selected. That does not rule out any other. There are numerous of hundreds of interactions here. I mean, you cannot describe it. It's very complicated. I read recently a paper which shows that in prebiotic experiments, simulation experiments, when you run the mass spec, you can see hundreds of species there, all kinds of different species there. Whatever you wanted, it's there. So, but this is not the way how you can solve the origin of life. Just if you submerge yourself in this super complexity, you're out. You cannot solve anything. And also, when you're looking, you're supposed to look something which resembles the modern biology. Simply because there is a rule of acceptance. You cannot, as I mentioned before, with the case with the self-replication and the case with the RNA world, as I mentioned, if something is very important at the beginning, it will stay important after. So it's not very easy to eliminate that.
Yeah, the only thing that's worth commenting on that is there are some people, and let me find the guy's name. Stuart Bartlett, for instance, is a person who deals with. I believe he's either deals with cell. I think he is a cell biologist. Yes, I believe he is. But he made the point a few years ago. I think it was a 2018 paper he published, kind of trying to discuss the fact that life, for instance, on planets like Encephalus or Europa or planets like that, they might have they might not have all the characteristics our life has. There could be other types of
Of course, as long
living
yeah.
systems.
Sorry that I disrupt you, but in fact, thinking if you don't have the feedback from the definition of life that I discussed, information, structure, and if you don't have the structure that harbor and preserve the information, you don't have life. You can describe any kind of system, but it's not life, because cannot progress, cannot go further. So yes, you can achieve this with whatever you want. I mean, I'm sure somewhere in another planet there might be conditions that you have RNA world completely based only on RNA and lipids, for example, but not proteins, another life. And that is completely feasible according to the description and according to the definition of life that I provided here. So you may have different kinds of forms. The problem with Europa, for example, Europa, is that if everything is submerged underwater, it's very difficult for me to guess how the polymerization might go.
Mm-hmm.
That's my problem there. And you need cycles, you must have a cycles, some sort of heat and cool cycles in order to get the dynamic. Without dynamic, it's very difficult to set up life.
Right, that's one of the interesting things as well. I remember reading a paper the other day. I believe it's Europa. It's however thick that ice layer is. It's like several hundred meters or something like that. But
there's a recent paper of the disciple of David Dimmer, Bruce Dahmen.
Yeah,
he's a guy that actually grew up with David Dimmer. Bruce, at the moment, is considered himself as a superstar in the origin of life. He published recently a paper that describes the possibility for life everywhere. He has kind of very sort of logical way of thinking. However, the point is not that you'll describe, for example, lipid vesicles. Yes, there might be lipid vesicles, but until you don't have a system where you have information and structure together, just this balance, you don't have advancing.
Right. Yeah,
There's
and
no
one
way
of
to
the
advance this.
yeah the curious thing with Europa is I believe a paper recently discussed that the ice layer there is actually relatively young. It might be only about like 100 million years old or something like that.
It
And
still
that's
might be very interesting. It still
Yeah.
might be very. I'm sure whatever it's there, it's interesting from perspective of science. It might not find the life, may not find any second origin of life, but it still will be very interesting. I'm entirely for this,
Yeah.
but I don't think it's these problems needs to be solved in terms of polymerization, the cycles, and so on. There is a cycles inside the actual Europa, because you have tides with the Jupiter. So you have tides
Right.
up and down, and that creates a heat.
Yeah,
And
it creates heat from gravitation.
yeah, gravitational heat. So let's go back to this particular picture. Short RNAs, and by saying short, I don't mean something more than 5 nucleotides. Very simple RNA. If you heat RNA or any nucleic acid, doesn't matter, RNA or DNA, if you heat it, it will melt. So you have a process of melting, separation between each other, and goes in a stage of single strand RNA or single strand DNA. And you can see here on the top, in a high temperature, there is just single strands RNA. And if you cool down, everything goes down, chill it down, they will start to interact naturally by base pairs. So Watson-Crick base pairs. And this is completely natural process, and it happens in every PCR cycle ever. So this is a process which I even don't need to argue that it's true.
Mm-hmm.
Simply, there's no other way. It is true. So also the process of amino acylation, meaning to attach for the ribose hydroxyl ribose to attach amino acid, it's a natural process. It happens very frequently. If you put them together, it will happen with some frequency. I wouldn't say very frequently, but with some frequency. So you have the process of up and down cycle with RNAs, and some of these RNA are amino acylated by nature in normal situation. So you don't want to create anything here. You just have this particular system. So, and when you go down in the temperature, chill down the temperature, you might have situation where 2 amino acids stay very close together, but they will stay close together not because just by chance they was close together, but just because you have a hybridization process between 2 amino acylated RNA. It's very simple, it's very natural, it's very understandable. Anyone problem with this?
Mm-mm.
So you have hybridization process, and this creates a situation where 2 amino acylated RNAs might bring very close together the amino acids. And naturally that might form a new peptide bonds between this. So by cycling up and down, up and down, up and down many times, you can expect formation of very short peptides. But this is not just peptides. This is a hybridization-dependent peptides. They depend from this hybridization. This is not smashed peptides. You can smash and create peptides just in solution if you put high gravity, high pressure, high temperature. They will join and you can have a short peptides, but this is not about short, just smashing peptides. It's a hybridization dependent peptides.
Mhm.
And that's natural. I don't see any problem, meaning it's hard to argue that this process might exist.
Mm.
Anyone to say something about that?
So basically the two nucleotides, or like a small polynucleotide, is amino acylated to a couple of amino acids and holds them together and allows them to polymerize.
Yes. So what you have two. Yeah, you said correctly. So you have two or more hybridized RNA. But in the site of this RNA might be amino acid.
Okay.
And that bring them close because you have hybridization process. So you hybridize, and it's happened that two of the amino acids comes very close.
Mhm.
And then they would polymerize.
And they polymerize.
Yeah. Okay. Good. Cool. I'm pretty busy looking at all the chats and the papers and stuff, but I'm so
Yeah.
I might miss a beat here. But
So let's go to the figure 2.
Okay. Figure 2. Okay.
Yeah.
Oh, let's see. It's the only thing I'm good for is scrolling.
Yeah.
Okay.
That's actually figure 2. It shows that you can expect all kinds of peptides to happen by chance in this way. So most naturally to think about is that most of the peptide will reflect exactly what is the abundance of amino acids, because if you have a process of prebiotic synthesis, not all amino acids have the same abundance. You have, for example, the glycine and alanine are hugely abundant, and all the next of the amino acids are indifferent. This is actually in the second paper described very well. So, and what you can expect, of course, the peptides that are formed, hybridization-dependent peptides, are formed simply because in the composition which reflects the actual abundance of the amino acids. You cannot expect anything else. So more abundant amino acids will be presented in these hybridization-dependent peptides more frequently. It is simple as that. So nothing surprising is having here. Now, if you go in the next figure 3, for example, yeah. So this is the picture which actually happened to be the figure 3 in the second paper as well. So the second paper also have the figure 3 and shows pretty much the same thing from another perspective. So these describing of by chance two different types of hybridization-dependent peptides where simply because of the presence of specific amino acid, you will have attraction to another amino acid, and I will describe that actually with the second paper. So let's go directly to the second paper at the moment, and that will be much more clear what actually happened. So
put that one up, get it sized.
I mean, this is well described. If you read the paper, you will, I think it's very clear what actually happened. But the idea is that to the attention, it goes, yeah, this is the second paper, and you can scroll down to figure, probably was 3. Figure 3 directly, not this one.
While we're trying to find something, RJ had a question earlier about the duplex codons. Let me see if I could pull it up
Is it
super
figure one?
fast.
Oh, we're on figure one.
Okay, so RJ says, question for Demeter. In due course, what is his view of duplex first idea of codons? So what do you think of the duplex first idea of codons, basically?
Duplex
I
first.
mean, so here is the important figure 3 and actually describes and explains everything.
Yep.
So if you think about in position 1, this is a hybridization peptide which happened by chance, purely by chance, initially by chance, which possess two kinds of amino acids. One is arginine and the another is aspargine. So those happen to be in two separate sites of this particular peptide. And you can calculate, I didn't do that exactly in this because the conditions are pretty much highly unknown. So any calculations for this particular probability will be off.
An estimate,
anyway.
yeah.
So, but there is a good chance in a peptide which contains a few amino acids to possess two types of amino acids like arginine from one side and aspargine from the another side. And one arginine, for example, it's very well proven, experimentally proven, that interacts with very good, this is the amino acids which interacts very well with RNA. And also not just very well, but interacts with GC-rich RNA mostly, I mean, with some high level than AU-rich RNA. So if you imagine interaction in a second stage here, in a second stage you have RNA and this bridge peptide interaction, but in the one side you have aspargine sitting there, which is negatively charged. By negatively charged, it simply attracts another arginine, which facilitates the amino acylation process very well. It's just increasing slightly, increasing and makes the way that amino acylation of this particular RNA, which is GC-rich because of the interaction of GC-rich arginine, it's slightly more likely to amino acylate arginine again. I have to ask here, is it clear? I don't like this question, is it clear? But now I have to ask. I have to ask because this is important.
Yeah. How the arginine like self-selects and stuff.
Yeah. So you have amino acylated in position 3 here. You have amino acylated RNA, which is happen to be GC-rich, and in the same time have arginine. This particular RNA, which is amino acylated with arginine and its GC-rich, will enter in a stage 4, which I just described, the process of up and down the natural PCR.
Okay.
And because it's entered there, it will increase very slightly, but will increase the probability, the bridge peptide, which is here in number 1, to come a little bit with higher probability. Because you already have a process of specific amino acylation of arginine with GC-rich RNA. So you have every cycle that you have this, every cycle increase slightly the probability of having bridge peptide like 1 here more frequently than the very first time, because you have stabilization, also you have stabilization process of this RNA. So the presence of this GC-rich RNA amino acylated with arginine will increase more and more every time. And that's the whole all about. So if you think about my first talk that I was talking about information and structure function, right? And the rule also. So how that happened in this particular figure? The information comes with the GC-rich RNA. It's a GC-rich. The bridge peptide actually, in fact, is the structure and function, and that determines it very well. I mean, not very well, but it's clear you have a link between the information and structure. How? Because you have process of more or less specific amino acylation of this particular RNA. So you have transfer of information from GC-rich to arginine containing peptide. And this is increased. Every single cycle, it's increased.
Cool.
So
system feedback loop.
this is by default, this system is first very simple. So simple. And the second, there is a transfer of information to some harboring structure or function.
Okay.
So a question that's interesting to me, at least, is that arginine is one of the amino acids that can be positively charged, but also another well-known one is lysine as well. Is there any conceivable mechanism for which lysine can interact similarly
It is
and increase its own?
Yes, in the paper, if you read the paper carefully, you will see there is a small section where it discusses the difference between lysine and arginine. Yes, it's absolutely sure any amino acids will be involved in this process. There's no doubt. And also lysine will be there as well. But arginine is much stronger. So you can think about this is a very small complex. It's vibrating very much with high frequency vibration. To stabilize this complex, you need much stronger interaction, which is arginine, not lysine. At the very, very first complex like this, which is so simple, you need something like more sure, more firm interactions to stabilize the structure. And that's why arginine, if you see the codon of arginine, the codon actually is GC-rich codon.
Mm-hmm.
And this paper shows that if you just order the codons from GC-rich to AU-rich, it's pretty much chronologically how the amino acids and the corresponding codons was assigned with the new codons chronologically. At the beginning, there was pretty much mostly RNA was GC-rich anyway. And this is due to many reasons which I described in the paper. Yes. So, and this is the main hypothesis which have description of many things. You can see how everything falls exactly in the spot in the process. So it's a simple system, a few amino acids and just short RNA. You don't need too much. It's feasible. There is no any single process in this particular mechanism where cannot exist. It's not proven experimentally somewhere and somehow in different types of experiments. All interactions are well known here. There is nothing that you can put the finger there and to say, oh, that cannot happen because of something. Of course, as a hypothesis, it's still hypothesis. That's why you need experiments. Yeah. So I will be back because of too much coffee. I'll be back in 1 minute, and we'll continue with Luca. I'll tell you a few things about Luca.
Okay. Luca and Origins of Life. Maybe I'll know what's going on.
Yeah, I was actually wondering about like he was mentioning the GC-rich interactions
Yeah.
that cause arginine to be essentially self-selective, like when it replicates. And what I was asking essentially is that is there like any known mechanism or plausible mechanism by which another positively charged amino acid, like histidine or lysine, could also like do that type of thing?
Mhm. Yeah, I don't think he quite answered. Did he misinterpret whenever I was asking about RJ's question with reference to like duplexes? Wait,
It could be he was. I'm not sure.
because I asked him about that, and then he went to the feedback loop and the amino acetylation and stuff. But that didn't quite seem like the whole wobble-based pairing idea, or like the wobble-based pairing developed after. Do you want me to repost it, or like repost the comment so that way you can pin it whenever he gets back and just have him?
No, it's fine. I think probably the best thing to do is probably like have him go like where he wants to in this conversation, and if he wants to have.
Yeah. I think we're going to have to read the papers again after this. I'm missing a few things because I'm looking at about 15 different things. Mm-hmm. But we have a ways to go to fully grok this, but this is pretty cool stuff.
Well, because there's a few things too. I mean, it might be that we have a hangout just going over like his papers particularly.
Right.
Because there's a few details there that are certainly interesting, like especially...
Yeah, and I want to look at that new book we just got on ribosomes. So,
yeah,
and a bunch of much more to do. There's always something to learn. Okay, you're back. You're back.
Okay, I'm back. Can you go to the first paper again? I'm sorry that this switching.
That's okay.
The first paper and go directly to the last figure, which is figure 4.
I wish Streamline was better at this, or StreamYard. Let's see, application window, and I'm looking at, God, which one is it? 2018, 2018. Got it. We're there, so you want figure 4?
Yeah, just go down to the figure 4. Now we'll dissect the Luca by itself.
Okay. Oh, is that 4?
Here we go. Yeah.
Yeah, it's not gonna. Okay, most of it. Yeah, it does. No, it doesn't all fit. There we go.
There we go.
How's that?
Yeah. So this is
a little bit bigger.
Yeah.
Okay.
So now, before starting with Luca, actually, any comments up to now? Just thinking about what it is. Questions about this mechanism. So, for example, where the energy comes from. This is one common question, because the metabolism first hypothesis, they always emphasize that they need to have some energy. But this is, I think it's not too much of a problematic question, because energy comes simply by when you have prebiotic synthesis of nucleotides and amino acids. They do not come in a clean form. They always come activated. They come activated by different another moleculars. They are not just clean nucleotides. They are not just clean form of amino acids. Simply the process of synthesis happen to be in a very harsh conditions, high temperature conditions with the different moleculars, And once they synthesize, they come with some activation of additional residues or charged additional forms. So, questions for Dimitar. What does suspect time frame earliest replication system? Okay, I will talk about the earliest replication system. Yes. So I will discuss that as well. And by the way, it will come very soon. So, so that is the answer for the energy. There is energy everywhere during that time, simply because of the prebiotic synthesis. And in fact, the developing of the metabolism is a way of independence. And this is a huge, powerful selection force. As much you pre-synthesize something as you are able to synthesize it by your own system, it means independence from the environment.
Mm-hmm.
So, for example, imagine that this system is now capable to synthesize its own arginine. For example, I'm just putting here an example.
Yeah.
If you are able to synthesize your own arginine, now you no longer depend of the arginine from the environment, which anyway is very low abundance. It's super low abundance. And now you are going forward, which is a huge selection force. So to me, developing of the metabolism is a way of independence from the environment. And that's how Luca became more and more and more independent from the environment by developing its own biochemistry.
Mm-hmm.
Also, that is absolutely sure you're supposed to have some sort of compartmentalization, some sort of separation. That's sure. There's no doubt. I mean, you have small pores, lipid vesicles, all kinds of those compartmentalization. And this is for sure should be there. There's no doubt. I'm not against those kinds of things. They have to be. You cannot have those system in an open space. So, question. Oh, no question. Okay.
Yes.
Now, let's go back to Luca. Here it's the evolution after figure 3. Whatever I just described as the bridge peptide system. What happened in the bridge peptide system, when I described in a figure 3, is the beginning of coding. As I mentioned, there was a information part and there was a structural part of the bridge peptide. So the non-coded HDP started to become actually coded HDP. This is a very sharp difference between chemistry and biology. It's not smooth in terms of understanding. You don't have a smooth transition from chemistry slowly going back to make a biology. No, at the same moment when you start to develop Darwinian evolution, which is figure 3 in the exact term, in exact mechanism, now you have Darwinian evolution kicks out because it's the same thing. The beginning of Darwinian evolution is the beginning of life. That's why you start up here, the beginning of Darwinian evolution, which is continue up to nowadays. From the coded HDP, hybridization-dependent peptides, of course, now because there is a Darwinian evolution, there is a process of increasing the complexity of the system. However, you cannot expect immediately to have a protein, long proteins at that moment. You have a very short proteins, very small peptides, actually not proteins, peptides. Now, here it's very important for everyone, and I don't know, everybody knows it, but somehow nobody actually pay attention. The difference between short peptide and protein is very simple. Short peptides interacts based on the amino acids that it's in this short peptide. If you have a long peptide, now you have a process of folding, of bending. In that case, you can create a catalytic center, which creates a new function.
Yep.
So, at the beginning, exactly at the Darwinian evolution, as I described, you cannot have anything more than short peptides, which function in one way or another way. One of the functions is, of course, RNA replication peptide, which facilitates replication, RNA replication, and that will give a huge Darwinian evolution advantage, selection advantage. But this is on a level of short peptides. At the very beginning, there will be just a few short peptides which will function. One function will be from the bridge peptide to make a specific coding for the specific amino acids. Another function will be in a way of RNA replication, and just a few more functions of short peptides. Most of the system will be RNA. This is resembles somehow the RNA world now, but it's not. It's not RNA world, it's RNA peptide world.
Yep.
And guess what? You have a huge limitation of functions of this particular very short peptides. They're limited in function. Therefore, you will select enzymatic activity from RNA. And this explains exactly why the ribozymes was selected. Because at that moment, you have only short peptides. You don't have formed long peptides that can form a bended structure, folded structure, and you cannot have catalytic centers yet.
Yeah.
That's why the protein development starts later, not exactly at the beginning. You have natural process of short peptide motifs, short motifs, slightly bended motifs, and then you have full process of protein formation. And at the same time, of course, RNA peptide world will be exactly until the first true proteins appeared. What mean true proteins? First to be folded, and second to have beginning, like start codon, and the end, stop codon. So if you have those two, you have real protein. And of course, this protein have a now more complicated 3D structure, which have a catalytic center, which function much more. The functions of catalytic centers and proteins are unlimited. You can have everything there. But this is not true for short peptides. They are very limited as a function, what they can do. From this figure, you can see at the same time when you have developing short peptides up to the real proteins, you have developing of ribozymes, and of course the developing of ribosome by itself. So, and that's it's logical at some point to finish with full formation of LUCA, where you have genetic code formation, you have proteins, you have RNA, you have DNA system, you have translation system, and then you have LUCA. So,
cool.
Anything that you want to discuss at that particular figure. So now you can go to second 2020 paper and show again the last figure 4 from 2020, and we'll finish. I will finish then for a large discussion.
Okay, let's see here. I'm moving a cat's butt around with my mouse. I got a cat on my. I got a cat problem. Okay, where do we want to go?
So go down to the figure 4.
Oh, aren't we? We're at figure 4. Oh, wait a minute. Okay, I'm on the
Yeah,
wrong
in
page.
this particular paper, yeah.
Hold on a minute. I gotta find it. Where did it go? History of the ribosome. I lost it. Sorry. Okay, I found it. Cat, get out of my way. Okay.
Some
I got a
people have cats, some people have parrots.
Yeah.
I have
I
my
got a
own
cat.
parrot here.
You have a parakeet?
Yeah, let me show you.
Oh God, I love birds. I wanted to get a pet parrot or parakeet or something, but I'm not in one place long enough to actually like have a pet that requires attention.
Say hi. Oops.
Aw,
Hi.
such a cute bird.
Here you
I got
are.
What an amazing little dinosaur right there.
Yeah, it is dinosaur. Yeah.
I got a big
Yeah.
lazy cat who keeps laying on my.
This is
laying
my
on my.
baby, which bothers me a lot. And it's very annoying every time because always ask for attention.
Yeah.
Yeah.
Yeah, that's what my cats. But both of my cats have been up here, and I've had to move my mouse. I have to move their butt when I move my mouse on the mouse pad. Okay, so we got figure 4.
So this is the figure 4, and this is a representation. Again, you remember the previous figure that I discussed about Luca, but he is more dedicated the part which is about the evolution of the codon formation and the amino acid synthetases. So I'm not going through entire paper because there are lots of things, but this is the arrangement of the GC codons to the AU codons, and you can see how the amino acids has been assigned during the entire process. And you can see how from bridge peptide there is a development from bridge peptide to the amino acid synthetases. However, there is 2 classes of amino acid synthetases which haven't been developed independently. They are not very similar to each other. They're kind of very different from each other. And you can see how naturally by stop codons you can separate the process of Luca formation in 2 phases. The phase 1, which I call pre-Luca 1, and phase 2, which I call pre-Luca 2. In pre-Luca 1, you don't have anything more than a few amino acids which are already assigned for codons. And this is the first, and this is the class 2 amino acid synthetases has been developed. And it's a very simple, pre-Luca 1 is very simple, but what is very important, the genetic code is formed by itself with the 3 nucleotide meaning genetic code. However, the 3rd nucleotide meaning, it's very loose, it's not very firm, but mostly on the first 2 nucleotide genetic code.
Okay.
Also, during that time, you have ribosomes, which has been developed, and the process of some sort of very loose replication. However, this replication, RNA replication, although the fidelity of this replication probably is very low, it's good enough to maintain the system. So you don't need to have high fidelity replication process in order to have this system to function, because you always have selection. Don't forget, it's Darwinian evolution. Always select and always select for the successful forms.
Yeah, that was something.
There are a lot. Yeah.
Oh, I was just saying the whole like wobble base pairing, like the wobble position developed after the initial like duplex of codon or duplex codons. That was something that I thought was really interesting that I just learned today. So
What I can say at the moment for the theory for codon formation and for the for evolution of genetic code, there's two streams of understanding of the codon of the genetic code formation. The first stream and all kinds of hypotheses, several of those, used to say something like this: because the codon is 3 nucleotides.
Mm-hmm.
With addition of every nucleotide, you add additional amino acids as an assignment. See what I mean?
Mm-hmm.
If you have just one nucleotide that is meaningful, you have just a few amino acids which is meaningful.
Yeah. So you think that
second nucleotide.
Mm-hmm.
Yeah, if you add second nucleotide which is meaningful, you just add more amino acids to
Mm-hmm.
this. And continue to the third nucleotide of the codon, and now you assemble all the amino acids. So this is the first style. Just to finish that,
okay.
This is the first style of thinking about the origin of genetic code, and I think this style of thinking for origin of genetic code is completely wrong.
Oh, okay.
It's not correct. What is to me and what comes with this model that I have here, it's totally different. You have all the 3 nucleotides codon formation, all the 3, in just a few amino acids.
Mm-hmm.
In the Luca 1. This is the second and the absolutely new, never published so far, new way of thinking for codon formation and for the beginning of the genetic code formation.
Cool.
Mm-hmm.
Okay.
So usually the first style that I described that it's wrong, they present the tables. You can look at. There is probably around 100 papers describing different types of tables, different selection forces that has been described.
Mm-hmm.
But unfortunately, none of those actually explain why, for example, certain portion of amino acid have a GC codons and another have a AU codons.
Mm-hmm.
This model that I published here explains it very well.
Yeah.
Explain all of that.
Cool.
So, do you think that the reason we developed the third codon, like the third base in the codon, I guess, was a response to third nucleotide. The third nucleotide in the codon was a response to a pressure to get more amino acids, or do you think it was added and then more amino acids became produced as a result of the ability to have more amino acids?
I vote for the second.
So it's very simple, and again, it is well described in the paper. However, I can simplify it very easily here.
Okay.
To assign new amino acid, you don't need new system of codon. To assign new amino acid for new codon formation, this amino to harbor, to take another codon combination, needs only 2 things. First, you need available codons, available combinations.
Mm-hmm.
And the second, you definitely need to have some function of this amino acid which select for. So, in other words, the new assignment comes because this amino acid was useful for something.
Mm-hmm.
But to be actually assigned for particular codon, this particular combination of nucleotides must be available. So not taken from the other before.
Mm-hmm.
So you have 64 combinations, and that's it.
Yeah.
Whoever take it, take it.
Okay, that makes a lot of sense.
Yeah. So
Okay.
I want to be sure that can you, Mike?
Yep.
Can you go over all the comments and just if there is any questions, someone that it's not answered, I just want to answer, because pretty much that's what I want to say today. However, we can talk a lot, but 2 hours is approaching, so I don't think it's.
Yeah. So I think James was asking particularly what the timing was for Luca, or what, how many years I think you think it was, or
Or like
do you?
an estimation.
Yeah,
So-
I don't know if that's a fair question.
Well, first of all, obviously nobody knows this answer, but I
Yeah.
can say something else. It's very interesting that if you look the fossil records, the origin of life should became very early, almost very early, after the proto-Earth was chilled down.
Yeah.
Mhm.
So I can say within the first, probably between 200 million or 600, in this period of time, between 200 million and 600 million years, you already have Luca.
Yeah.
So that's also very interesting, and that says something else. And I never seen comments on this. This says that Mother Nature tell us something and tell us how probable the life is to start.
Yeah.
Answer is, according to the Mother Nature, will be very highly probable event.
Yeah.
Okay.
Why? You have life immediately after Earth formation. You can imagine another scenario. A scenario where all the fossil records didn't show any form of life in the first 2 billion years, for example.
Mhm.
You can imagine this scenario. It's possible, right? So the fact, if you have such a scenario, you can think for some like a panspermia. You can think about something like, for example, very rare event.
Yeah.
Yeah.
But the fact that it started so early, meaning that whatever you're doing, whatever you're thinking for origin life, should be extremely simple and probable.
Yeah.
And
I
by
think
the way, if you see this hypothesis, you have a few amino acids and short short RNA.
Mhm.
So what more simple than this you can have?
so. We got a question here. Yeah. Anyway, I think it's inevitable, and I think it's basically built into the orbital structure of carbon, hydrogen, nitrogen, oxygen, etc. That these things are going to form in these complexes if we have an environment like the Earth with our temperature range, that sort of thing. That it's inevitable, and we're going to find it everywhere. So Otangelo has a question.
I hope.
Otangelo is not our friendliest.
He seems like a nice guy.
No, he's a really nice guy, but he's probably not on our side, is what I'm trying to say.
Oh yeah, he's definitely not. He's
Definitely not on our
great.
side. So anyway, since when has chance the urge to select or okay? Really, Otangelo, we need better questions. We need more specific questions. So you can let that one go, or you can respond if you want.
How to produce cipher? What do you mean by cipher?
Or to put, they're talking about genetic codes and information and all that. There's all this hullabaloo about information, and I've looked into it, and it's wonderful. Biosemiotics and all the stuff that, those papers from the Royal Society. There's a lot of wonderful information in there, and none of it says that there was an intelligent designer. They're trying to understand the complexity, how complexity works. And and these guys tend to read it as, there must have been an intelligent designer. Well, nowhere in there do I see an intelligent designer. I see these atomic, tiny little systems forming together and creating more complexified systems. So basically, my God is complexity. I love complexity. I sit around with Legos and combine them in different orders, So
But think about, you're right. So think about, to have more complexity, you must have Darwinian evolution set up.
yeah,
Otherwise,
exactly.
otherwise,
Yeah,
every
like,
time when you lose this particular system, next time you'll come with the same probability. You will not have increasing the probability to have something more
right.
complex. So
Yeah, it basically creates its own probability wave ahead of it, or
exactly.
it spells the probability wave.
And this is the huge problem in most of the modern hypotheses that I've been reading, and I read a lot hypotheses. So they simply usually order whatever they think happened in some sort of chronological way, but from the complexity from simple to complex.
Yeah,
But they do not explain the actual mechanism of selection.
right.
You don't provide
Do
what
you like this
actually
guy?
is the mechanism of selection, you don't have good hypothesis. So I know this book, Nick Land.
Yeah, yeah. Do you
Uh,
like? I like Nick Lane. He had a lot of really good ideas, but I think this is from, when was this written? The Vital Question. I can't see the copyright. 2015. I guess that's pretty new.
so
Anyway.
this is it is hydrogen vents,
Yeah,
hydrogen
yeah.
vents hypothesis. So the
Yeah.
main concept and idea here is you have alkali conditions inside the hydrogen vents, and you have acidic conditions outside of the hydrogen vents, which creates a proton gradient. So the proton gradient goes from high acidity to low acidity, which triggers lots of new synthesis of new forms, of new species, of new more complicated forms. So let me say, first of all, there was experiments and they didn't show any significant gradients. That's one thing. But the second problem in all of the hydrogen vent hypothesis is exactly what I mentioned about the cycles, that you need to have wet and dry cycles. Otherwise,
Yeah.
you don't have polymerization. And that's a huge problem. If you don't have polymerization, whatever you're doing, you don't have much complex there. And that's one thing. I completely agree. The hydrogen vents, they might be very useful to produce organic moleculars. Absolutely, no doubt. I mean, yes, they might be one of the main, plus the volcanic eruptions, main producers of organics, which is a wonderful, good thing to think about prebiotic synthesis, but this is not life.
Yeah.
To have a life, you must have Darwinian evolution. Without Darwinian evolution, there's no life. This is the same thing as I started to say. So,
Okay. Well, I think we want to read your papers again and do a little homework, and maybe revisit all of this. And when do you think your new paper will come out?
I'm going to submit that sometimes after September.
After September.
Between September and the end of the year, I still don't know exactly when.
Okay.
But the manuscript is ready, and this paper actually contains exactly the description of life, what the Darwinian evolution is, and the law for origin of life.
Okay.
So the law of origin of life, I'm repeating it again. The information, the structure, and the rule between them are locked in its origin.
Yeah. And we wouldn't mind if you shared that with us in advance. If
exclusive tape.
I can share it. Yes.
Yeah, and we will not share it any further. We'll just study it. So anyway, okay, cool. So okay, is there anything
also
else?
any questions? If in the after reading the papers, and you can pretty good. I mean, any kinds of papers, not necessary to be just this my papers, but anything else with that it's just puzzling you or trying to figure it out or thinking. You can point me a paper that I don't know. That's for sure. It's very interesting as well.
Yeah, there is one paper that was very similar to yours, and I can't remember where I put it, but I want to find that and go over that again, because it was talking about some of the same things you were talking about with the connection between the nucleotides and particular proteins.
Probably you're talking about De Faras. This is the Brazilian scientist. So
okay, it might have been, yeah.
The Faras, actually, they do have a very similar way of formation of hybridization peptides, very similar to what I'm describing, but it's not the same because they do not provide the mechanism as a bridge peptide that I provided here.
Okay, so could you spell that name, please?
De Faras.
Dave.
I think this is what it come in my mind, De Faras. Actually, you can find in the literature of this particular paper.
Like in your references?
Yeah, in the reference, yeah, you can find the Faras, and you can probably all see it. And I also described the paper. This is, but the problem with the hypothesis, it's all about the selection between specific sequence of RNA and specific amino acid. If you don't have this link somehow, specific amino acid with specific sequence, you don't have transfer of information.
Okay, I'm trying to find it.
I think it's source 13, De Faras ST, evolution of transfer RNA and the origin of the translation system.
13. Okay, got it.
Yeah, that's it. These guys, and he have many. He published several papers actually.
Okay, so anything else? Should we end this and chat for a few minutes afterwards?
If anyone have something to comment or questioning, I'm fine to close now, so it's okay.
Okay, how about you two, Andy?
We have a contact with you by email. If you're always welcome to send me emails and asking me, or I can ask you, or whatever. We can set up another discussions anytime.
Sure.
That's fine.
Okay. Let us do our homework, and maybe we will have more highly intelligent questions for you later.
No.
My low brain evolutionary biology mind is.
Yeah, that's okay. So
Okay.
it's not much. Yeah.
We got a lot of highlights in your papers, and just putting the whole thing together, and the notes I have here, I want to basically let it all come together in my mind. But hearing you talk about it was very, very helpful. So
I hope you figure it out what I mean. The most important thing is it's simple.
Yeah.
It's a simple system. And the second one, it's explain a lot. You have explanation why the ribozymes was selected. You have explanation why the genetic code started to be. You have explanation what is the information and how simple the transfer of information goes to the new protein. Everything goes smoothly well. And I in this second paper, there's so many coincidence that fit very well to the theory that you can check it out from the nature. So I hope, for me personally, I hope very soon I'll set up some experiments with my buddy that I published this paper. But I don't know when I can do some experiment. What the experiment will be just to show appearance of the hybridization dependent peptides. So we'll try to figure it out how to make hybridization peptides exactly as I described in figure 1. This first paper. So I'll try to make it in tube. This.
Okay.
James has a question for you.
Hi,
What's-
James. I'd
wants to know
like
what
to
you
know
thought.
what Dimitar thought of the book appendix. Well, so I don't know how time I will have, but definitely that's fine if you add appendix to the origin of life. Should be
Yeah.
no problems. It's the only thing. It's the time what I have
Mhm.
and how busy I will be. That's it.
Yeah. Okay. So let's end this unless anybody
Okay.
else has anything.
Nope.
You guys, Andy, Walker, you good?
Yeah, I'm good. I don't think Immutable's still here, or Andy's still here.
Oh,
I think, well, he said he had to run take care of something. He's been in the chat, but he's been muted on the stream for a bit.
Yeah.
okay. Okay, so I'm going to end the broadcast. We'll see you guys, and
Okay.
stick
Bye-bye.
around for a minute.
Yeah.
Stick around, Demeter, for just a minute. And broadcast.